Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Imagine having a superpower. Like, you can speed up evolution by a truly massive amount, maybe 100,000 times.
Wow.
And not over millions of years, but in just days.
Yeah.
Right there in a lab.
Okay, that's already pretty wild.
So here's the really strange part. This super fast evolution, it only hits the specific DNA you want it to.
Mm-hmm.
The rest of the cell's important genetic stuff, totally untouched. Safe.
Right, so targeted hyperspeed.
Exactly. So today, we're diving deep into this groundbreaking new system. It really promises to change how we engineer proteins, how we tackle big problems like antibiotic resistance.
That's right. Yeah, our mission today is to unpack this new paper introducing T7 Oracle.
T7 Oracle.
Yeah, it stands for T7 orthogonal replisome-assisted continuous laboratory evolution. Quite a mouthful.
It is.
But it's basically this powerful platform they built in E. coli, the workhorse bacteria,
Okay.
and it just fundamentally changes the game for how we can do directed evolution. So we'll dig into, how it's built, its design, how they tweaked it really carefully, and what it could mean for, well, everything from new drugs to better industrial enzyme.
Okay, let's unpack this then.
Huh.
Because usually when you think evolution, you think slow, right? Gradual changes,
Mm-hmm.
huge timescales.
Millions of years, yeah.
But in the lab, if you want to, say, evolve a protein for a new job,
Mm-hmm.
maybe make an enzyme work better or something, you need mutations.
Right, mutations are the fuel.
But the big problem has always been, if you ramp up the mutation rate too high in a living cell,
Mm-hmm.
well, you tend to kill it.
Mm-hmm. Damage the essential genes.
Exactly. Its own crucial RNA gets messed up. So scientists have dreamed for ages about finding a way to speed up evolution, but keep it targeted.
Keep it contained.
Yeah, a way that doesn't break the whole cell.
Exactly. And, historically we've had ways to make mutations, like
Right.
error-prone PCR in a test tube, or chemical mutagens.
Outside the cell, mostly.
Right. Or people tried engineering the cell's own DNA copy of machines, the polymerases. But you hit this basic limit, you just can't mutate the host's own genome too much, or it won't survive.
It's too risky for the cell.
Totally. Now, there were systems in yeast, like OrthoRep, that proved you could have a separate, isolated replication system, like a genetic engine running alongside the main one.
Okay, so the concept was there.
The concept was proven, yeah. But yeast isn't E. coli. E. coli is what most molecular biology labs use day to day.
It's the standard.
It's the standard. And the earlier tries in E. coli, things like EcoRep, they just didn't get high enough mutation rates, or they weren't stable enough.
Didn't quite cut it.
Didn't cut it. So the real GAT, the thing people wanted, was a system in bacteria that could handle massive mutation rates but keep it safe in a sort of sandbox. Something that worked with standard lab stuff and could run continuously.
Yeah.
That's why getting this robust, insulated system working in E. coli, well, it's a really big deal.
That context definitely helps frame it. So, okay, how did they build this sandbox, this orthogonal system inside the E. coli? What are the parts? What were the clever sort of engineering tricks they used?
Well, the big idea, the conceptual leap, was pretty neat. They decided to engineer the whole replication machinery, the replisome, from a bacteriophage called T7.
A virus that infects bacteria.
Exactly. Think of it like importing the virus's entire DNA copying engine plus its support crew and getting it to run as a second independent system inside the E. coli.
Wow, okay.
So they put in 4 key T7 genes, GP1, GP2.5, GP4, and GP5 using plasmids. Those are just small circles of DNA.
Standard tools.
Standard tools, yeah. But a really crucial tweak involves something called T7 lysozyme.
Okay.
Now, normally it helps break open the cell for the virus, but it's not strictly needed for replication itself. They found, though, that sticking an engineered version of it, one that couldn't break things, a hydrolase-deficient one, onto the T7 RNA polymerase, that was somehow key to kicking off the replication.
Interesting. A non-obvious connection.
Yeah, a bit unexpected. They also used a specific starting point for T7 replication called OAR, and they began with a T7 DNA polymerase that was already a bit sloppy.
Sloppy how?
It was exonuclease deficient. Basically, it lacked its natural spell checker, its proofreading function.
Ah, so it makes more mistakes, more typos.
Exactly. Which
Now,
is just what you want if you're trying to speed up evolution by introducing mutations.
right, okay, that's quite a setup, building this viral engine inside a bacterium.
Yeah.
So once they had this intricate sandbox put together,
Mhm.
what was the first big test? The proof it actually worked.
Yeah, good question. They really needed to show two main things. Was it stable, and was it truly isolated?
Did it last, and did it keep the mutations contained?
Precisely. And the results were pretty great. The T7 Oracle strain was remarkably stable. It kept going for over 2 weeks.
Wow.
And it grew really well. Doubling time, just 29 minutes, which is fast even for E. coli. And it reached high cell density.
So it wasn't making the cells sick.
Not at all. But the real kicker, the aha moment, was the mutation rate. On the plasmid, with the T7 origin, that's where you'd put your gene of interest, they measured a mutation rate of about 3.3 times 10 to the minus 8 substitutions per base.
Okay. Which is higher than normal.
It's significantly higher than the background rate. But here's the crucial part. They checked the host cell's own DNA, its main genome.
Yeah.
Its mutation rate stayed incredibly low, tiny, like 3.6 times 10 to the minus 10, orders of magnitude lower.
So the sandbox walls held.
Absolutely. That confirms the orthogonality. The mutations were completely stuck on the target plasmid, leaving the host genome untouched.
That's huge.
It really is. And one more thing, the system was fantastic at taking up new circular plasmids. Transformation efficiency was super high, like 2.4 times 10 to the 10 colonies per microgram of DNA.
Which means it's easy to use in the lab.
Exactly. Much, much easier than systems that use linear DNA. Very user-friendly.
Okay, so they've got isolation, robust growth, a decent mutation rate, and it's easy to work with. But I bet decent wasn't the end goal. You mentioned hypermutation.
Right. They wanted to crank the dial way up.
So how do they take this already pretty cool system and push it even further? How do they tweak that T7 DNA polymerase to make it really error-prone, but like in a smart way?
Yeah, that's where they really got into the fine-tuning. So remember, DNA copying accuracy or fidelity usually relies on three things.
Okay.
How well the polymerase picks the right base to add, that proofreading function we mentioned, the exonuclease activity.
Spell checker.
The spell checker, yeah. And then the cell has its own backup systems called mismatch repair.
Right.
So the first polymerase they used, the 28 one, already lacked proofreading. That alone gave them about a 25-fold jump in mutations compared to the normal T7 polymerase.
Okay, step one.
Step one. Next, they went after base selection itself. They focused on the polymerase's active site, the part that actually grabs the DNA bases. They made libraries, trying out different changes at 11 specific spots in that region. And they found 3 key single-letter changes, specific mutations: N520M, P560V, and V443K.
Okay, just 3 specific tweaks.
3 specific tweaks. And when they put all 3 together into one polymerase, the triple mutant, boom, the mutation rate shot up
How high?
to an incredible 1.04 times 10 to the minus 5 substitutions per base pair.
Whoa. Okay, that's a huge jump again.
Massive jump. But they didn't stop there. They did more random mutagenesis, just throwing mutations at the polymerase and screening for even higher rates.
Pushing it further.
Yeah, and they found 2 more helpful changes, Y24F and Q585R. Put those together with the first 3, you get the quintuple mutant.
5 changes in total.
5 changes. And that one hit 1.73 times 10 to the minus 5 substitutions per base pair.
Okay, put that number in perspective for us.
Right. So compared to the E. coli's natural mutation rate in its own genome, this is about a 100,000-fold increase.
100,000 times faster.
Yeah. Confined only to the plasmid they want to evolve.
Oh.
It's like going from walking pace to, I don't know, warp speed for discovering new molecular functions.
That's just mind-boggling speed.
It really is. And another important point, the type of mutations the supermutator polymerase made was pretty balanced. It wasn't heavily biased towards creating just one kind of change.
Which is good for evolution.
Crucial. You want diverse mutations to explore different solutions, not get stuck down one path just because your mutator prefers it.
Right, you need variety. Okay, this speed, this precision, it's honestly amazing. But the big question, the one I know everyone listening is thinking, is does it actually work in practice?
Does it deliver? Yeah.
Can this crazy hypermutation actually solve a real problem? Like, say, the antibiotic resistance you mentioned earlier. What did they test it on?
Yeah, they picked a perfect target for a real-world test. an enzyme called TEM1 beta-lactamase.
Okay, what does that do?
It's a very common bacterial enzyme. It's a major reason bacteria become resistant to antibiotics like penicillin and related drugs, the cephalosporins.
Ah, one of the bad guys in resistance.
A key one, yeah. So they used T7 Oracle to evolve this enzyme continuously. The goal was to make it better at breaking down newer, important antibiotics used in hospitals, drugs like astronam, cefotaxime, and ceftazidime.
Okay, so evolving resistance in the lab, basically.
Exactly. They set up 96 parallel experiments. Think of them as 96 little independent evolution universes running side by side.
Right.
And they grew the bacteria, passing them to fresh media under higher and higher concentrations of these antibiotics,
Turning up the pressure.
turning up the pressure. And get this, they reached concentrations up to 1,000 times higher than the starting point in just 6 days.
6 days. That's it.
6 days.
Wow. And what happened? Did they see resistance evolve?
Oh, yeah. The results were pretty stunning. They saw this amazing convergence. The same key mutations started popping up across many of the different parallel experiments.
So evolution found similar solutions independently.
Exactly. And here's the really cool part. Almost all the mutations they found had already been seen before,
Seeing where?
either in actual drug-resistant bacteria isolated from patients in clinics or in previous, much longer lab evolution studies.
So this super fast system rediscovered clinically relevant mutations.
Precisely. And what really jumps out, as you said, is the speed. This single one-week experiment basically summed up findings about these types of resistance enzymes, extended spectrum beta-lactamases that took researchers decades to piece together before.
Decades of work. In a week. That's almost unbelievable. It kind of makes you feel for all the researchers who spent years on this before.
Right. It's revolutionary, but yeah, maybe a little humbling too. And they didn't just see the end result, they tracked the mutations appearing daily. They could literally watch as mutations like, say, G238S and E104K, which help resist cefotaxime, appeared and then took over the population. Or R164HS for ceftazidime resistance.
So you see the evolutionary trajectory in real time.
You're watching evolution happen on fast forward. It's incredible.
Yeah. Okay, but this raises a really interesting point about evolution, doesn't it? Organisms, or enzymes in this case, often find the easiest path, right? The path of least resistance on what scientists call a fitness landscape. They might find a peak, a good solution, but maybe not the highest peak, the absolute best solution.
That's a classic challenge in directed evolution, absolutely. You can get trapped on these local fitness maxima. You find a solution, maybe quite a good one, but maybe there's a much better one you missed because you started from just one place.
Right. So how does T7 Oracle help with that? Does it just find the local peaks faster, or can it explore more broadly?
That's where another key advantage comes in. Remember how easy it was to get plasmids into the system?
Yeah, the high transformation efficiency.
Exactly. That means you can easily start your evolution not just from one single starting gene, but from a whole library of pre-made variants.
Ah, so you start with diversity already built in.
Precisely. You can integrate things like site saturation libraries directly into this continuous evolution process.
Okay, site saturation library. What's that?
Imagine picking a few really important spots in your protein, say, in the active site of the enzyme. A site saturation library is where you systematically create variants that have every possible amino acid at those specific spots.
So you try all the options at key locations.
You try everything at the critical positions. So they did this for TEM1. They made a library covering 5 key active site residues.
Okay.
Now, when they started evolving from 96 individual clones picked from that library, they saw lots of different solutions pop up. Lots of different ways to improve the enzyme.
Showing there are many paths.
Many paths, yeah. But here's the really neat part. Then they tried starting the evolution with the entire library mixed together as one big pool.
Okay, throwing everything in at once.
Right, and what happened? The mutations in the active site converged again. Evolution found preferred solutions. But they were different solutions than what they got when they started just from the original wild-type enzyme.
Different and better.
Different and more active. It strongly suggests that by starting with that much broader exploration of the possibilities.
Giving evolution more starting points.
Exactly. T7 Oracle could find those global maxima. The truly optimal solutions, not just the first good one it stumbled upon.
So it helps you find the highest peak in the whole mountain range, not just the nearest hill.
That's a perfect analogy.
Yeah, it's really powerful. Combining hypermutation with smart library design.
It really is. And what's kind of fundamentally groundbreaking here is that T7 Oracle isn't just a one-off trick. It's a general platform It's E. coli native, it runs continuously, and it has minimal hassle in terms of workflow.
Easy to adopt.
Yeah. Compared to older systems like the yeast orthorep or another technique called PACE, this T7 Oracle system is just simpler. It doesn't need really specialized or expensive equipment.
So most labs could potentially use this.
That's the idea. It makes this kind of power much more accessible.
Okay, so let's bring it back to the listener. What does this all mean for, the future of science, maybe even for us?
Well, think about it. This technology basically turns making libraries of molecules from a static one-time thing into a dynamic living process.
Like evolution as a service.
Exactly. Evolution as a service. And the fact that it's so robust, remember the host genome is safe, they got rapid results on a real enzyme that makes it really credible for people to actually use it widely.
So it speeds up the whole discovery cycle.
Massively. That design, build, test, learn loop that drives science, this compresses it. Instead of months or years, maybe you're talking days or weeks for some steps.
Which could mean faster development of what? New medicines?
Faster therapeutics, definitely. Better biosensors, maybe for diagnostics. And crucially, like we saw, creating rapid countermeasures against things like antibiotic resistance. It lets us potentially keep up with how fast pathogens are evolving.
Keeping pace with nature's evolution.
Trying to, yeah. Now, are there still challenges? Of course.
Like what?
Well, people will want to fully map out if there are any subtle biases in the types of mutations it makes, even if it looks unbiased now. And testing how well it handles really, really big pieces of DNA might be important for some applications.
Ensuring stability with large constructs.
Right. But the potential for scaling this up looks huge. You could imagine running multiple different evolution experiments in parallel, maybe automating the whole culture system.
Wow.
It really could accelerate the pipelines for developing new biologic drugs or industrial enzymes. It helps democratize high-throughput protein engineering, bringing it to more labs everywhere.
Not just the big specialized centers.
Exactly.
Okay. So as we wrap up, maybe a final thought for everyone listening. Thinking about this power, this T7 Oracle system, if we can now harness evolution, literally direct it, to engineer biology at this incredible speed, what completely new things might become possible? What possibilities open up for life, for technology that maybe we haven't even really started to imagine yet?